I'm a basic scientist who's interested in understanding the fundamental biological processes that underlie the development and the progression of multiple sclerosis, with the hope that we can identify specific molecules or pathways that can be targeted for therapies. And as Pam alluded to, multiple sclerosis is a disease where our immune system attacks our own brain and spinal cord. There's no cure for the disease, and Alberta happens to be one of the highest rates of incidence. So the specific question we're interested in asking are there endogenous protective mechanisms in multiple sclerosis? And it's a very simple idea that we have behind this. So whenever our bodies encounter an injury, whether it's a bruise or a cut, our bodies mount a natural healing mechanism. And in MS and other diseases, it's likely that healing mechanisms come into place, but it's obvious that they're not enough to overcome the disease. So the questions we're asking is, can we identify these natural healing mechanisms in MS? Can we figure out why they're unable to overcome the disease? So are they inhibited or defective in some way? And then can we harness the protective functions to overcome the disease? So one of the molecules that we work on is something called alpha-B-crystalline that we identified a few years ago that showed to be actually protective, that it prevents your immune system from becoming activated and it protects your brain cells. And we tackled our experiments in a very multidimensional way. We use animal models, cell culture, cell and molecular biology techniques. But one of the reasons why I came to the Hotchkiss Brain Institute a couple of years ago is that we had the opportunity to actually collaborate with some of the members of the MS clinic at the Foothills Hospital. So in addition to our animal studies, we can actually obtain blood samples from patients and isolate their immune cells and ask whether any of these molecules we identify are able to beneficially alter the function of these cells. And the reasons why we do this is that multiple sclerosis is a very heterogeneous disease. It seems that every single patient has a slightly different disease. And it's unlikely that one single therapy is going to cure the disease, which is we already know from the present drugs out there. And so what we need to do is identify all these possible protective molecules and all the other ones that are identified by other collaborators around the world, and maybe we use them in combination to specifically target subsets of patients. So the basic science is really important for us to understand how these molecules work and be able to collaborate with our medical colleagues to be able to translate this in our labs. So thank you.